Mm-hmm.
We'll just give it another 30 seconds or so before we start.
Sure. So this is going to give me the impression that I'm presenting to 3 people, because I can only see the 3 of you.
Whether that
makes the nerves.
Yeah, I won't tell you. Yeah.
Yeah, no, don't tell me. That's the one advantage of these Zoom presentations. But I don't know if it's an advantage. I do like being in the same room as people, and I do like, because this format where people ask their questions and a moderator has to repeat the question sort of removes a bit of contact with the audience.
Exactly.
But we'll see how things pan out in the future.
Exactly. So on that note, we are at 1 minute past the hour, so I think it's a good time to get started. I'll just take the opportunity to welcome everyone. We're going to hear from Anne-Noël Samaha about addiction-relevant behavior and how the pattern of drug intake is relevant for establishing addiction or maladaptive behaviors relevant to addiction. And before I turn it over to Ankush to introduce our speaker, I'll just mention that you can leave your questions in the Q&A using the Q&A feature or using the chat. And as always, feel free to sign your questions so we can get to them at the end and know who's asking. So Ankush.
Hello and welcome everyone for today's event. And today we have Dr. Samaha here. Dr. Samaha was born in Dubai and raised in Lebanon and in Canada. She is a behavioral neuroscientist interested in the neurobiology of reward and drug addiction. She completed her PhD at University of Michigan with Dr. Terry Robinson. Her dissertation received the James McKean Cattle Award from the New York Academy of Sciences. Dr. Samaha completed her postdoctoral training at Center for Addiction and Mental Health in Toronto with Dr. Kshitij Kapoor. In 2008, she joined the faculty at University of Montreal, where she is now a full professor. Using animal models, she is examining how temporal dynamics of stimulant drug use, continuous or intermittent intake, fast or slow drug onset, determine outcome. Her work has earned awards from the ACNP, SFN, and a Senior Investigator Award from the Canadian Society of Pharmacology and Therapeutics, the highest honor in pharmacology in Canada. Dr. Samaha has been the FRQS Research Scholar since 2009, and her research is funded by the Canadian Institutes of Health Research, the Canada Foundation for Innovation, and NSERC. Aside from science, Dr. Samaha loves bicycles, good food, and good books. So over to you, Dr. Samaha.
Okay, thank you very much, Ankush, for the introduction. I'm going to share my screen with you and program my pointer. Voila. Okay, so it's an honor for me to be here to present the lab's work to you, especially when I see the list of other panelists, a very prestigious company I'm in. So for a number of years, my lab and I have been studying the effects that drugs of abuse can have on the brain and on behavior using animal models. And we study stimulant drugs specifically, namely cocaine. And one effect that cocaine can have is to produce addiction. The common belief is addiction develops in vulnerable people when they take large amounts of drug, because the assumption is that exposing oneself to large quantities of drug is both indispensable and uniquely effective in producing the changes in the brain and psychological function and in behavior that underlie the transition to addiction. But my lab has been discovering surprising things about what it takes to develop the patterns of drug use that are characteristic of addiction. And we're finding, using our animal models, that how often drug is taken can be more important than how much in predicting outcome. And the outcome we're interested in, of course, is addiction. And in this context, animal models of voluntary self-administration of drugs are really quite useful, and I would even say very precious, in isolating the drug-induced changes in function that promote the development of addiction. And of course, when we want to model addiction in the laboratory, we want animals to do more than just take drugs. We want them to show the features that are relevant to the disorder. And so what we look for in these animals is quite similar to what people with addiction can show. So a high motivation for drug, continued drug seeking despite adverse consequences, a high risk of relapse after abstinence. And in this context, a really significant advance came in our field in 1998 when Serge Ahmed and George Koob developed what came to be known as the long access procedure. So this involves allowing animals to self-administer drug almost continuously for extended daily sessions lasting 6 or more hours. And if you estimate brain cocaine concentrations during such a session, you'll see that brain cocaine levels will rise at the beginning of the session and remain quite high for hours on end. So, because when you give animals almost continuous access to drug, they will take it almost continuously. And long access animals are very often compared to short access animals who are also allowed to take cocaine or any other drug continuously, but during shorter sessions lasting 1 to 2 hours a day. So not surprisingly, if you compare average daily drug intake between the two groups, the long access animals are taking quite a bit more drug. But most interestingly for researchers in addiction, the long access animals are more likely to also show features that are relevant to addiction. So all the features I showed on the previous slide, they are willing to work much harder to obtain drug, they escalate their drug use over time, they are more vulnerable to relapse, they incubate their drug craving after abstinence. And so a key assumption behind the use of the long access procedure is that the animals are showing these features because they are taking high levels of drug. However, the long access procedure, while it might model a cocaine taking binge that some human users might engage in occasionally, it doesn't model the temporal pattern of human cocaine use, which is typically intermittent, both between and within bouts of use. So we know that in typical users, there will be periods of cocaine used and then periods of non-use and so on and so forth. And even during a bout of consumption, cocaine users tend to take the drug intermittently, presumably having brain concentrations rising and falling between hits. And there was this also very interesting laboratory study published a number of years ago where they allowed experienced cocaine users to take IV infusions of cocaine in the lab. And they realized that the participants would regularly forego a dose. And when asked why, they replied that they could get a better effect from the next dose if they skipped one once in a while. So even experienced cocaine users themselves can clearly see the advantages or benefits of intermittent cocaine use. So in the context of this literature, Dave Roberts and his then student Benjamin Zimmer decided to model intermittent cocaine intake in laboratory rats. And they did something very clever. So they allowed the animals to self-administer cocaine during extended daily sessions, just like in long access. The difference being that cocaine was only available in discrete trials. So during 5 minutes, the animals could lever press and get cocaine. But then during 25 minutes, the levers would retract and the animals could not get any more cocaine, and so on and so forth for the 6-hour session. And if you look at a typical pattern of intake in an intermittent access rat, well, this is it right here. So each tick mark is an injection that the animal actually took during the session. And intake is necessarily intermittent because that's how the session is programmed. And you can see that this would produce spikes and troughs in brain cocaine concentrations during the 6-hour session. A pharmacokinetic profile that is really quite different from what you can see in a typical long access rat. So these rats will generally take cocaine almost continuously throughout the session, and this would keep brain cocaine concentrations quite high throughout the session. If you measure intake over days, not surprisingly, it's quite a bit higher in the long access rats than it is in the intermittent access rats who are down here with some other control groups. And in fact, intermittent access rats take about the same amount of cocaine as short access rats do. But the really surprising findings came when Zimmer and colleagues used behavioral economics metrics to assess the willingness to work to obtain cocaine. And they saw that responding was highest in the animals that had taken cocaine intermittently, even though these animals had taken much less cocaine than the long access rats. So this was really revolutionary in our field because it suggested that intermittent cocaine use could be more effective than high and sustained levels of intake in producing change in drug use over time. And the surprises kept coming because a couple of years after the initial publication, there was additional work to suggest that intermittent versus continuous cocaine intake might produce opposite effects in the dopamine system. This is work conducted by Erin Calipari when she was a PhD student in Sarah Jones's lab, also working with Dave Roberts, and she is actually next week's speaker. So what was done here is 4 groups of animals were generated, a cocaine naive control group, and then intermittent access, long access, short access rats that could take cocaine under these different conditions for a number of sessions. The authors then performed fast scan cyclic voltammetry recordings in the nucleus accumbens core to assess the ability of different concentrations of cocaine here to block the reuptake of dopamine. And they found something rather surprising. In the long access animals down here, there was a reduced ability of cocaine to inhibit dopamine uptake. In the intermittent access, they saw quite the opposite. Cocaine was better at blocking the inhibition of dopamine uptake. And this same result is shown in a slightly different way here, where I'm showing you Ki for cocaine. So this is a concentration of cocaine that was needed to produce 50% of dopamine uptake. You can see that you needed less cocaine to produce this effect in the nucleus accumbens core of intermittent access rats, but you needed more cocaine to produce the same effect in the long access rats. So in sum, long access to cocaine promotes tolerance to the drug's effects at the dopamine transporter, while intermittent access promotes sensitization to cocaine's neurochemical effects. So these types of findings are fueling ongoing debates about the role that hypo versus hyper dopaminergic states play in addiction. And a great coincidence, just last week, we published an opinion piece on this very topic. So if you're interested, you can read it. Now, to get back to this notion that high and sustained brain levels of cocaine are necessary to produce changes in drug taking and drug seeking that define addiction, well, a former PhD student in the lab and I, Florence Alain, were interested in examining this dogma a bit more closely. And in a first series of experiments, she compared these 2 groups of rats. So both would take cocaine intermittently, meaning they would have daily 6-hour sessions where cocaine was available in discrete periods, separated by longer timeout periods. And the difference between the 2 is that this intermittent access group could take as many injections as they wanted during each drug available period. In contrast, this limited group here could only take 4 injections. And so we reasoned that if high levels of cocaine intake are necessary to increase the motivation to take drug, then later on, these animals would respond more for cocaine under progressive ratio than these animals. And for those of you who are not familiar with progressive ratio, it involves making it such that animals have to respond an exponentially greater number of times on the lever or any other operandum to obtain each successive injection of the session. So just to show you estimated brain cocaine concentrations in a typical intermittent access limited versus unlimited rat. So we were achieving spikes and troughs in brain cocaine concentrations under the 2 conditions, and the peaks would be higher in the unlimited animals because they can take more cocaine during each drug available period. And this shows you intake across the 10 sessions. Down here are the limited animals, and this arrow is just to remind you that they could only take 4 injections during each drug available period. They generally took all 4, and intake was rather low and stable across the sessions. The unlimited animals took more cocaine, and they also escalated their intake across sessions. So by the end, cumulative intake was quite a bit higher in the unlimited group. But when we allowed them to respond for cocaine under progressive ratio across a range of cocaine doses, the 2 groups of animals worked equally hard to obtain cocaine. So the amount of cocaine taken in the past doesn't predict the motivation to obtain the drug in the future. To address this further, Florence generated 2 new groups of animals. Now we had a group of long access animals. So these guys had a 6-hour drug available period every day. And these down here are the intermittent access limited animals. These animals had intermittent access to cocaine. And in this cohort, we limited the number of injections they could take even further. So here they could only take 2 injections during each drug available period. So again, if the amount of cocaine taken in the past determines the later motivation to work for cocaine, then the long access animals should show higher breakpoints for cocaine under progressive ratio. So again, just to remind you that the 2 procedures would produce very different pharmacokinetic profiles in the brain. The long access rats do indeed take cocaine almost continuously for 6 hours. This would produce high and sustained brain concentrations of the drug, while in our intermittent access limited animals, we have these small peaks and troughs in brain cocaine concentrations throughout the session. Here are the intake data during the 10 daily self-administration sessions. So quite low levels of intake in our intermittent animals because they only have 2 injections to take during each drug available period. And the long access animals really take a lot of drug, and they escalated their intake, such that by the end of these 10 sessions, the long access animals had taken about 7 times more cocaine than the intermittent access rats had. But look at what we found under progressive ratio. So again, across a range of cocaine doses, the intermittent access animals worked significantly harder for cocaine than the long access rats, which again supports the idea that more cocaine taken in the past does not necessarily predict more motivation for the drug in the future. So a couple of conclusions from this first series of experiments. We saw that with intermittent cocaine intake, high and escalating levels are not necessary to increase responding for the drug. We saw that if cocaine intake is intermittent, even quite limited exposure to the drug would be effective in producing this addiction-relevant change. Now, another dogma in the addiction literature is that you have to give animals extended daily sessions to produce changes in drug use that are relevant to addiction. And this is such a widely held belief in our field that manuscripts and grants get rejected sometimes if you propose to use shorter self-administration sessions. And I know this because it's happened to me. So Florence and I decided to also examine this widely held belief a bit more closely. And so we wondered whether with intermittent access to cocaine, do we still need extended self-administration sessions to produce addiction-relevant behaviors in our rats? So to address this question, here we had 2 groups of animals. They would both take cocaine intermittently, and the only difference is that these guys had 6 hours a day to self-administer, and these guys had 2 hours a day. So somewhat of a copy-paste of the original short access, long access comparison, but here under intermittent access conditions. After a number of such sessions, we assessed responding under progressive ratio. And in this experiment, we also looked at relapse-like behavior using the reinstatement model. So this was reinstatement of drug-seeking behavior triggered by an IP cocaine injection. Okay, so this is the number of injections the animals took across the 18 sessions, low and stable levels of intake in the animals that have 2 hours of intermittent access to the drug every day. But the long intermittent access rats took a lot of drug and escalated their intake. So up to this point, the findings map on quite well to what you would see if you compare traditional short access, long access rats. And so by the end, obviously cumulative intake was highest in the group of animals that had 6 hours a day to take the drug. Now, in this experiment, we looked at measures of intake, but we also looked at cocaine-induced locomotor activity during each of these sessions. Cocaine is a psychostimulant drug. It it enhances psychomotor activity, and in both laboratory animals and humans, with repeated exposure to the drug, you can see psychomotor sensitization. And the reason why addiction researchers like myself are interested in psychomotor sensitization is because of the idea that it results from some of the same forms of drug-induced neuroplasticity that also produce increases in drug wanting over time. So here are some locomotor activity data, but not just locomotion. So you can see these are findings in the animals that had 6 hours a day to take cocaine. These are findings from the 2 hour a day group. So you can see that if we estimate brain cocaine concentrations in these animals, the peaks would be higher on the last session. And this makes sense because they actually took more drug by the end than they did at the beginning. But the animals in this group remained stable in their intake. So the peaks didn't change over time. And then when we looked at locomotor activity, this was quite cool because it also followed a spiking pattern. And this is interesting because if you look in the literature, typically the temporal pattern of cocaine-induced locomotion is really tightly coupled to the temporal pattern of cocaine-induced increases in dopamine concentrations in the striatum. So we reason that we're probably not just producing a spiking pattern in locomotion, we're probably also a spiking pattern in extracellular dopamine in the striatum. And we can see here that compared to their locomotor response on the first session, on the last session, there was an increase in locomotion, and this could suggest the development of psychomotor sensitization, but we can't be sure because they also took more drug on the last session, so they could just be locomoting more because they are taking more drug. But this was certainly not an issue in these animals here because they didn't escalate their intake across sessions, and so you can see that their locomotor response to cocaine was greater on the last session than on the first, suggesting that these animals had developed psychomotor sensitization. Now to go back to some self-administration data. So this is what happened when we put the animals under progressive ratio so they could earn different injections of different cocaine doses, and across all doses, we saw no group differences. The two groups worked equally hard for cocaine. Then we gave them a 6 hour extinction session. So this involves putting them back in the cages. They can lever press, but they don't get any cocaine. And we can see that drug seeking responses are quite similar in the two groups. And then to reinstate drug seeking behavior, we injected all animals with an IP injection of cocaine. They all reinstated and there were no significant group differences. So long and shorter intermittent access sessions produced similar levels of incentive motivation for cocaine and drug primed reinstatement. So we saw in this experiment that only rats given long intermittent access sessions escalated their cocaine use. However, rats in both groups increased their locomotor response to cocaine over time. The two groups also showed similar breakpoints for the drug and similar levels of relapse-like behavior. Again, hammering the point that how much drug is taken in the past doesn't necessarily predict the response to the drug in the future. And so it appears that with intermittent cocaine intake, extended exposure is likely not necessary to model in rats the changes in brain and psychological function that are involved in addiction. Now, the intermittent access procedure is still relatively recent, and so we have to keep in mind certain considerations. Intermittent intake during each session is imposed, it's not chosen. And this is quite different from what you see in the real world, where people can determine the temporal pattern of cocaine intake. And during each session, rats don't have access to alternative rewards. And this is a limit of the procedure, but it is being remedied by work in different labs, including Yavin Shaham's lab. And intermittent intake so far has only been studied in rats. We have no idea of the types of effects it could have in non-human primates. And also, we still have very limited information about how useful intermittent access self-administration procedures might be in identifying new treatments for addiction or mimicking existing treatments, what is referred to as reverse translation. So the intermittent access procedure is a model to study addiction in the laboratory. And the question, well, it's inherently imperfect, as all models are, but the question is it useful to study addiction in the laboratory? And the answer is yes, absolutely. So if you follow the literature, you can see that there are increasing publications with the procedure. Ever since the initial work of Zimmer and colleagues, papers are coming out showing that intermittent access to cocaine increases the motivation to take the drug, that it promotes continued drug use in spite of punishment, and that compared to continuous intake, intermittent intake of other drugs of abuse also promotes addiction-like behaviors. And more recently, there was work coming out to suggest that intermittent intake of cocaine might not only promote increased drug wanting, but it might also promote the incentive motivational effects of drug-paired cues. So there's work showing that intermittent cocaine intake is especially effective in promoting cue-induced reinstatement of cocaine-seeking behavior and incubation of cocaine craving. Now, drug-paired cues play a very important role in drug addiction. They can invigorate drug-seeking and drug-taking actions. They can evoke craving and relapse after abstinence. And they can also come to be pursued in their own right. So they can become condition reinforcers. And this is something that we saw in David Belin's talk last week. And so with the postdoc in the lab, Sean Cue, we wondered whether taking cocaine intermittently versus continuously might allow cocaine-paired cues to acquire greater condition reinforcing properties. So Sean designed his experiment in 2 steps. So first, he had 2 groups of animals taking cocaine, either intermittently or under long access conditions. So they would lever press, get a cocaine injection, and each injection was paired with a light and a tone. So these stimuli became cocaine-paired cues. On test day, the levers were reconfigured in the test cage, and now the animals could lever press to get the light and tone stimulus alone without cocaine. So if they do lever press to obtain these cocaine-paired cues, we conclude that the animals have attributed motivational value to these cues because they can act as condition reinforcers, and then we can compare the 2 groups. So just to show you some intake data. So this is what the animals did during the self-administration sessions. They had 10 sessions. Here I'm showing you lever presses. Here I'm showing you number of injections earned. So as we've seen before, the long axis animals press more because the levers are available continuously during the whole session. And they also take more drug. And here we also had a new control group. So NC is for no cocaine. So these are animals that were put in the cages every day, but they were not self-administering cocaine. They were just being flashed with the light and tone stimulus. So this was just to control for any intrinsic effects of the light tone stimulus. By the end of these 10 sessions, the long axis animals took more drug. And in this experiment, it's particularly important to note that this means that they were also getting significantly more cocaine cue pairings. Now, 3 days later, the animals go back in the cage, and now they can press one of 2 levers to get the cocaine-paired cues alone, without cocaine. And so what we saw is that compared to the cocaine-naive control group, the 2 cocaine-experienced groups did press more on the levers, and they did in fact press to obtain cue presentations, as shown by significant lever distinction and discrimination, sorry. And what we saw is that responding was highest in the intermittent axis animals, meaning that these animals earned more cue presentations during the session than the other groups did. Sean then waited 30 days after the last self-administration session and repeated this test. And by this point, the long axis animals were just no longer interested in working for the cues, but the intermittent axis animals were still going strong, as you can see here by group differences in the number of Cue presentations. So these are brand new data and they're still preliminary, and we're determining the extent to which they can replicate. But these data certainly suggest that rats attribute more incentive motivational properties to cues paired with intermittent versus continuous cocaine intake, and that this effect is longer lasting also in the intermittent access animals. So in plain words, this means that when cocaine is taken intermittently, all the discrete environmental cues that are paired with cocaine use can acquire greater control over behavior and also more persistent control over behavior. Now, in the last chapter, experimental chapter of this talk, I'd like to share with you some newer data where we use the intermittent access procedure to examine pharmacological treatments for cocaine addiction. Now, those of you in the field will know that there are currently no medications approved to help people with cocaine addiction reduce their cocaine use. But a really promising strategy is D-amphetamine maintenance therapy. So this involves giving amphetamine continuously in a slow release formulation to reduce the potential for abuse. And it actually works. So in both humans, in non-human primates, and in rats, amphetamine maintenance therapy can reduce cocaine seeking and taking behaviors. And the question is, how does amphetamine do this? One possibility is that amphetamine interferes with cocaine's effects at the dopamine transporter, because I've shown you that one thing that cocaine does pretty well in the brain is to block the reuptake of dopamine, thereby increasing dopamine neurotransmission and producing reward. And there is certainly support for this idea that something's happening via the dopamine transporter. So this is work done by Siciliano and colleagues, also in Sarah Jones's laboratory, where they compared two groups of animals that were allowed to self-administer cocaine for 14 days. And one of the two groups was maintained on amphetamine treatment during this phase. So they were implanted with an osmotic mini pump that contained amphetamine. And so for 14 days, they were co-treated with amphetamine. The amphetamine treatment was then stopped. And then using behavioral economics metrics of the motivation to take cocaine, the two groups were tested early on and then 7 days later. And what they observed was that the amphetamine decreased later responding for cocaine. And they also looked at changes in cocaine-induced inhibition of dopamine uptake. And this was also ex vivo in the nucleus accumbens core. And they first replicated their previous finding, showing that in animals that had taken cocaine under long access conditions, there is tolerance to cocaine-induced inhibition of dopamine uptake. And what was truly interesting here is that in the animals that had also taken cocaine under long access conditions, but that had been co-treated with amphetamine, this is the curve in blue, cocaine-induced inhibition of dopamine uptake was perfectly normal. So this was interpreted to suggest that amphetamine reduces cocaine use by preventing tolerance to cocaine's effects at the dopamine transporter. However, as you'll have noticed here, the animals were given cocaine access during long access self-administration sessions, and this doesn't model the temporal pattern with which humans take cocaine, which tends to be intermittently. And also, I showed you earlier in the talk that long access versus intermittent access to cocaine can produce completely opposite effects at the dopamine transporter. Tolerance in one case, sensitization in the other case. So given these things, we set out to determine how amphetamine maintenance therapy might influence both cocaine taking and using behaviors and also cocaine-induced inhibition of dopamine uptake at the dopamine transporter. It took many people to do this. So Florence Alain led the project, did all the behavioral experiments along with Marie-Pierre Baudouin, and Benoît de Lignan-Lavaux and Eric Trudeau did all the voltammetry recordings. And my colleague Vincent Jacquemet extracted dopamine uptake inhibition data. And Terry Robinson helped us interpret and write the manuscript, interpret the results and write the manuscripts. So you'll notice here the symbol for males, because I'm first going to show you how amphetamine influenced cocaine use in males, and then I'll show you the same effects in females. So here we had two groups of animals, both taking cocaine intermittently, and one of them was co-treated with amphetamine. And this is what we saw when we looked at intake across 14 self-administration sessions. So amphetamine treatment didn't significantly change ongoing cocaine intake, but it did prevent the development of psychomotor sensitization. So we measured locomotor activity during each session. It increased across sessions in the control animals, but it did not increase across sessions in the amphetamine co-treated group. And this is further highlighted down here, where I'm showing you locomotor activity across the 5-hour sessions on the 1st, 7th, and 14th day. And what we also found here, we're very interested in discovering that amphetamine not only prevented the development of psychomotor sensitization, but it also flattened the spikes that are normally seen in locomotor activity when an animal is taking cocaine intermittently, which suggests that maybe amphetamine is also flattening spikes in extracellular dopamine concentrations in the striatum. So after the initial 14 sessions, we stopped the amphetamine treatment and we compared the groups on responding for cocaine under a progressive ratio schedule. And then if you pay attention here, you'll see that this group, after progressive ratio testing, was split in two. Some of the animals went on to be tested for extinction and reinstatement, and some of them were used to address a different question. Here, what we did was we allowed the animals to self-administer more cocaine, but this time with amphetamine co-treatment. And we did this because in the real world, people who present at an addiction clinic for help are necessarily already cocaine experienced. So we wanted to see whether amphetamine treatment could reduce cocaine using behaviors in animals that already have a history of taking cocaine. Okay, first I'm showing you what happened here. So when we put the animals under progressive ratio where they could lever press for cocaine, we saw that the amphetamine pre-treatment reduced responding for the drug. This is further highlighted here where I'm showing you cumulative cocaine intake during each progressive ratio session at the different cocaine doses. You can clearly see that the amphetamine pre-treated animals were just not interested in working that hard for cocaine. When we put them under extinction, we also saw that amphetamine reduced cocaine-seeking behavior under extinction conditions. And amphetamine also reduced cocaine-primed reinstatement of drug-seeking behavior. And note that the reinstatement test here was now 2 weeks away from the end of amphetamine treatment. So this suggests that amphetamine could reduce cocaine-seeking and taking in a very persistent way. So its therapeutic effects could outlast its presence in the body. So now I'm going to take you to the second group of animals that were put back were given intermittent access to cocaine once again, but now with amphetamine treatment. And then we compared their responding for cocaine under progressive ratio after this amphetamine treatment versus before. And we saw that after treatment, the animals responded less for cocaine than they had before. And I'm showing you here the individual data points at each cocaine concentration before and after amphetamine treatment. So amphetamine co-treatment also reduces the motivation to take cocaine in animals that already have a cocaine taking history. Very important from a translational perspective. Now I'm going to show you what amphetamine does in females. And when we started this work, Aissatou Ndiaye, who's a PhD student in the lab, who led this work, her and I went through the literature to see, has anyone specifically analyzed the effects of amphetamine on cocaine-using behaviors in females? And if you know of any such work, please do email me, because we weren't able to find any specific analyses in females. And so it was important to do this given that addiction afflicts both women and men. So we want to see how treatments influence cocaine-using behavior in both sexes. So she essentially replicated the previous experiment, but in females. So here are intake data in the two groups. In blue are the animals that are being co-treated with amphetamine. And you can see that amphetamine didn't significantly change ongoing cocaine intake. So by the end, cumulative intake was quite comparable between the two groups. We also looked at psychomotor activity in this experiment as well. And you can see that in the animals not treated with amphetamine, locomotion follows a spiking pattern, and the animals are also developing psychomotor sensitization, while in the animals co-treated with amphetamine, the spikes in locomotion are flattened, and we don't see a significant increase in locomotion, so no evidence of psychomotor sensitization. Now, after these sessions, animals responded for cocaine under progressive ratio. We tested them then under extinction and reinstatement. And we found that amphetamine reduced responding for cocaine under progressive ratio. It reduced responding under extinction in the females as it had in the males. But in the females, having been treated with amphetamine didn't influence cocaine primed reinstatement. So save for this exception here, generally speaking, amphetamine maintenance therapy is effective in reducing cocaine seeking and taking behavior in both males and females. So it has similar effects in the two sexes. So now we're going to go back to the males so I can show you the last piece of data where we assessed the effects of amphetamine maintenance therapy in animals that were taking cocaine intermittently, and we assessed effects on the dopamine transporter. So specifically the ability of cocaine to block dopamine uptake. Well, I should explain the groups. So we had two groups taking cocaine intermittently, one co-treated with amphetamine, and then we needed cocaine naive rats in this experiment. So these are two groups self-administering saline, one of which was co-treated with amphetamine. So this is all happening ex vivo in the nucleus accumbens core. Using fast scan cyclic voltammetry, we found that, first of all, in the presence of different concentrations of cocaine, when we looked at stimulated dopamine overflow, there were no group differences. So it didn't matter if they had taken cocaine in the past or not, if they had been treated with amphetamine or not. So we see no group differences. And this is further highlighted here, where I'm showing you the two cocaine experienced groups. So these groups here, as a function of percent of the saline control animals, which we had just pulled together because they were no different. And so no effects on stimulated dopamine overflow. But there were group differences in cocaine-induced dopamine uptake inhibition. So this is shown here, and you can see the effects of different concentrations of cocaine on dopamine uptake inhibition. And what the first thing to note is that here is a replication of the original finding of Calipari and colleagues, showing that in animals that have taken cocaine intermittently in the past, this enhances the drug's potency at the dopamine transporter, and there's enhanced dopamine uptake inhibition. However, this is completely prevented if you co-treat the animals with amphetamine while they are taking cocaine. This is shown in a slightly different way here, so you can see enhanced cocaine potency at the dopamine transporter prevented by amphetamine co-treatment. So to conclude from this last series of experiments, amphetamine treatment during intermittent cocaine intake reduces the motivation to seek and take cocaine in both sexes. It was efficacious both in previously cocaine naive rats and in cocaine experienced rats. And we saw that intermittent cocaine use promotes sensitization to the drug's effects at the dopamine transporter, as shown previously by the Calipari paper. And the novel finding here is that amphetamine treatment reverses this effect. So amphetamine treatment might reduce the cocaine taking and seeking behaviors normally produced by intermittent access by preventing sensitization-related changes in cocaine potency at the dopamine transporter. So as general conclusions to bring all this work together, the temporal pattern with which cocaine levels rise and fall determines the risk of developing addiction-relevant behaviors. Intermittent cocaine intake, even with very limited exposure, is really uniquely effective in producing psychomotor sensitization, incentive sensitization, dopamine sensitization, and consequently, addiction-like behavior. And finally, I really believe that self-administration procedures that reflect the intermittency of human drug use could be ideally suited to study the addiction process and to pilot new treatments. So on this note, I would like to thank the very hardworking people in the lab that continue to do this work. Florence is not in this picture because she's off doing her postdoc. She's gone from the lab. She's in Brigitte Kiefer's lab. And I really owe a great deal to the students in the lab because these are very labor-intensive experiments. as all experiments are, and they work very hard. And so I'd also like to thank the funding agencies for making this work possible. Thank you for your attention.
Wonderful. That was a really thought-provoking talk. And I see we have a couple questions already in the chat. And if we have time at the end, I'll ask mine. So, Ankush?
Yeah, so Dana Collins asked, did you see differences in responding in the no drug periods between the limited intermittent access rats and unlimited intermittent access rats? What about between the long intermittent access and short intermittent access?
So if I understand the question correctly, is what do the animals do in the timeout periods, correct? If we see differences in responding during the periods where they get no drug, it's kind of difficult to assess because we retract the levers. So it would be interesting to find some way to see whether would the animals seek drug and the patterns of seeking during this period, but we retract the levers so they can't even press them anymore. So we don't really know. Would the limited animals be showing you like a frustration effect where they would go crazy on the levers during the timeout periods? And also we'd have to think of ways of doing this because the most significant cue to the animal signaling that this is a no drug period is retraction of the lever. So we just take them away and the animals understand that, okay, now this is a timeout period.
So I had a question. So you mentioned that 2 injections of cocaine in the intermittent access are willing to, the mouse would be willing to work harder. Is there a kind of threshold, as in if it's 1 injection, would that behavior change?
Are you asking, how these animals respond to a single drug challenge?
Yeah.
For example, so yes, so Terry Robinson with his student Alex Kawa did some work to that effect where they took intermittent access and long access and short access rats and then just challenged them with a single cocaine infusion. And they found that the animals that had intermittent access history showed a sensitized dopamine response to this single cocaine infusion. So again, evidence that intermittent exposure to cocaine really promotes sensitization-related neuroplasticity.
Thank you. So Rustinal asks, does intermittent access only enhance motivation for drug rewards? Long access procedures do not seem to dramatically affect natural reward seeking, but I have not seen comparable data comparing drug and natural reward seeking with intermittent access.
Okay, so in this, to answer this question, I have a cool paper to recommend to you, published by my lab. This is Gaye and colleagues, published in Neuropsychopharmacology, where we looked at how intermittent access to cocaine versus food pellets would influence incubation of cue-induced reward craving. And we found that there was no significant cue-induced food craving after intermittent access to food, but there was very significant cue-induced craving for cocaine reward in animals that had taken the drug intermittently.
So thank you. And Robinson Mike asks, beautiful talk, Anna. It seems that the principal effect of intermittent access is to sensitize the response at DAT. What adaptations might be happening in situation of intermittent access to other drugs that have different modes of action, such as heroin?
Yeah, that's an interesting question. So there's a really important literature, not to be ignored, showing that all drugs are not the same. So there is a work by Aldo Bariani, among others, showing that essentially that all the experiments we do, for example, with cocaine, we can't take for granted that the same neurobiological principles would hold with drugs of other classes. So I think this is what we know for now is that since there have been intermittent access studies with other drugs of abuse, including heroin, and we can see that intermittent access to heroin is also equally effective in promoting addiction-relevant patterns of heroin seeking and taking behaviors, just like initially seen with cocaine. But the intermittent access procedure is still relatively recent. We're getting a greater comprehension of its behavioral effects, but a lot of work still needs to be done to understand how it's changing the brain.
So I have a question, which is both amphetamine and cocaine have effects on striatal dopamine, right? So do you think that is a way by which it's mediating its effect in decreasing the cocaine use?
Okay, I have a hypothesis. A hypothesis that you can sort of see here. So what is amphetamine doing in the brain to prevent the cocaine-induced plasticity at the dopamine transporter? One possibility is that when animals take cocaine intermittently, you would be producing these spikes and troughs in brain dopamine concentrations in the striatum. And what amphetamine would be doing, because amphetamine also promotes dopamine release and inhibits the reuptake of dopamine. So what amphetamine is likely doing is producing sort of a increased tone, increased dopaminergic tone in the striatum, such that if on top of that an individual is taking cocaine intermittently, the peaks and troughs would be less pronounced. And so why is this relevant is because we know from an older literature where experimenters would inject animals with drugs that intermittent dopamine stimulation with dopaminergic drugs is really very effective in producing sensitization-related neuroplasticity. And here what we're doing, perhaps by giving amphetamine, is we're flattening these peaks, we're flattening these intermittent spikes in dopamine, and maybe that's a mechanism that contributes to the eventual decrease in drug seeking and in motivation for cocaine.
So that brings me to another question, which is, given this amphetamine treatment, what would happen if I stop it suddenly? What kind of effects do we see? Do we see relapse?
If we were to stop the amphetamine treatment suddenly, well, this is what we did here. So in these experiments, what we did was we treated the animals with amphetamine while they were self-administering cocaine, and then we stopped the amphetamine treatment before measuring responding for cocaine under progressive ratio, extinction reinstatement. So all of these metrics were taken after the cessation of the amphetamine treatment. And so I know that in clinical trials in humans, what people generally do is look at how ongoing amphetamine treatment is influencing cocaine using behavior. But the animal studies, and this is not the only one, there's previous work by Dave Roberts and others showing that, and also the Siciliano and Al paper also showed that if you stop the amphetamine treatment, the therapeutic effects of amphetamine go on, they're persistent. So it's not like you stop the amphetamine treatment and then the animals, this enhances cocaine seeking behavior. It's quite the opposite. The decrease in cocaine seeking continues after the cessation of amphetamine treatment. The question is, how long does it continue? And this I don't know.
Makes sense. So Morgan James says, "Hi Anna, lovely presentation. I am interested in your thoughts on escalation of intake. You versus others see under intermittent access conditions. How does this square with sensitization of effects at the DAT?
How does this, I missed the end.
How does this square with the sensitization of effects at the DAT?"
Okay. I'm not quite sure there is quite a good relationship or between escalation of cocaine use and the development of sensitization-related neuroplasticity, because we did a bunch of correlational analyses and we saw that the extent of escalation does not predict the extent or the robustness of psychomotor sensitization. I also showed you in the study where we compared longer intermittent access sessions versus shorter intermittent access sessions. In the shorter sessions, no escalation is possible. Yet these animals showed significant psychomotor sensitization. And so I would predict that you would also see sensitization to cocaine's effects of the dopamine transporter, even if animals don't escalate, because we have many pieces of evidence showing that escalation is probably separate and different from the plasticity that produces increases in psychomotor activity and increases in cocaine-induced dopamine uptake inhibition.
Okay, so we have a question from an anonymous attendee who asks, what's known about the mechanisms of tolerance versus sensitization of dopamine transporter function? Is the pharmacokinetic profile that causes the difference as suggested by the AMPH data? And if so, would non-self-administered intermittent cocaine also cause these changes in dopamine transporter function?
Okay, we do not know, because to my knowledge, this has not been addressed. So if we were to just, instead of allowing animals to self-administer cocaine, if we just gave them the cocaine ourselves along with the amphetamine treatment, would we prevent any effect of experimental administered cocaine? But it's important to note that work has shown that in order for amphetamine to reduce responding for cocaine, the cocaine has to be taken voluntarily. So just exposing animals to cocaine alongside amphetamine doesn't produce the same effect as animals that are actively taking the drug. And so this addresses another possible explanation, effectively excludes the possibility that it's a pharmacokinetic interaction, because the animals have to self-administer the cocaine for you to see reduced cocaine using after amphetamine treatment.
So yeah, go on, Megan.
We have 2 more questions in the chat and 4 minutes, so we'll try to get to the last 2 questions before we hit the hour.
So another anonymous attendee asks, says, "Nice talk. What would be your hypothesis regarding the effects of KOR antagonists on cocaine breakpoints following intake? Core antagonists have been shown by Cook and colleagues to attenuate cocaine breakpoints following long access conditions."
Okay, so I know that people have looked at how. The short answer is we don't know. So I don't know of any data where this has been assessed, specifically comparing long access to long access to intermittent access. But manipulations of kappa opioid receptors do indeed influence cocaine using behaviors. But how this might pan out in the long access versus intermittent access conditions, we don't know.
Okay, so Jackie Meh asks, I hope I'm pronouncing the name right. They ask, given the high rates of circadian rhythm dysregulation in drug users, do you believe the time of the day that intermittent access paradigm occurs impacts behavioral outcomes and motivation?
The time of the day. There is a literature out there to show an influence of the circadian rhythm on the response to cocaine. So why not? I mean, I don't think this has been done, but I think it's quite possible, because the response to drugs of abuse can vary throughout the circadian cycle.
I had a question regarding how differently does amphetamine and cocaine cause dopamine release? As in, what are the rates? Is cocaine higher or amphetamine higher?
Well, generally speaking, amphetamine is produces a greater increase in dopamine, extracellular dopamine concentrations in striatal areas. And so the textbook answer is that cocaine blocks the reuptake of dopamine, and this is how it increases dopamine neurotransmission, while amphetamine not only blocks the reuptake, but will actually trigger release via changes in the conformation of the dopamine transporter. That's the textbook answer, but there is evidence to suggest that cocaine as well can directly promote release of dopamine. But still, if you look at, for example, in vivo microdialysis data, the increase in extracellular dopamine concentrations is really significantly higher with amphetamine than it is with cocaine. Now the question is, how do you equate doses? And that's a whole other ballgame.
So Megan, you had one question?
I did. I mean, we're right at the hour. I just thought it was, really interesting thinking about, and you touched on it regarding the natural rewards or the patterns of heroin intake, and thinking about things like prescription opiates that have extended release or long axis release, where, the plasma levels are quite stable, but the pattern of actually taking the drugs could still be intermittent despite the long pharmacokinetic profile. Just thinking about the implications of, I don't really have a specific question since we're at the hour, but thinking about how these factors might play in with the behavior of the taking the drug being intermittent, but the plasma concentrations staying high in these extended release formulations, and what implications that might have.
So if I understand you correctly, you're wondering whether, for example, people who take methadone, this is generally taken orally, and the kinetics are quite slow and sustained, but the intake is relatively intermittent. So there you would expect a bit of a drop in methadone plasma levels before the next dose. And these are all unanswered questions, because what constitutes intermittent? And my instinct tells me that based on the data that's being collected in the field, that you need sharp peaks and troughs to promote the plasticity that then facilitates increased drug seeking and drug wanting. That's what my instinct tells me, and we'll see if history says I'm correct or not.
Wonderful. So we are just over the hour, so I think we will wrap it up. And as Dr. Samaha already indicated at the beginning of our talk, next week we will hear from Erin Kalapari. Thank you again.